Hello and welcome to another one of these ADD videos. Today we're going to be looking at membranes. Membranes make up the outer barrier of the cell, and they're made up of these phospholipids here. Oops, way up here. The phospholipids have two parts to them. They have a hydrophilic head. That means it likes water and is usually charged, and a hydrophobic tail. And that means that it doesn't like water, and it's usually made of carbons and hydrogens. Okay? And what happens is they form a bilayer that separates the outside here from the inside of the cell here with a bunch of these phospholipids. All right? And basically, in a neuron, what they do is they try to create these gradients using things like sodium potassium pumps and just resting permeability to potassium ions. We'll talk about all that later. But basically, it establishes a concentration gradient in which there are high concentrations of ions outside the cell, specifically the sodium, calcium, chloride, small proteins are high outside and low inside. Okay, and what that does is it creates a natural tendency for these ions to want to come inside there. And the same is true for potassium on the inside, which is high outside and low it's high inside and low outside. So its natural tendency will be to efflux or get out of the neuron. So this roughly establishes your resting membrane potential at negative 65 millivolts. And I will be going through that on an entire different video dealing with the potential, action potential, and stuff like that, okay? But in these membranes, you can have these proteins embedded in them that have different functions. The first type of protein that you might find in them is an ionotropic protein. And this ionotropic protein is a channel, basically a pore, that in response to a trigger, something either binding to an active site of the channel or a voltage change would be another trigger. Some of these channels are voltage dependent. What happens is the trigger, whatever it is, opens the pore. And once it opens, it can either let stuff in or out depending on what the gradient is. Again, it's always going to go from a high concentration to a low concentration. That's the principle of diffusion. Okay? Another advantage of the ionotropic type of signaling to a cell is it's more of an immediate cellular change. These ions coming in the cell are going to cause instant changes. Whereas a metabotropic is more of a second messenger system, meaning that when you activate it, it activates other things and ends up, either activating transcription factors or opening internal calcium reserves. But either way, it goes from an inactive state up here, and once it either binds a ligand or it gets activated somehow, one of the subunits of this G protein, the actual second messenger, goes off and just phosphorylates stuff and just wakes it up, dental cyclase cascade or whatever it is. It starts just doing things. But the thing here you got to notice is that because it's activating many things that they in turn will activate, it's a delayed response. It's not instantaneous like opening up a hole in the cell, okay? So that's the main difference between metabotropic and ionotropic. Then there are symports and antiports. What symports do is they use ATP, common source of energy in your cell, right? When you cleave it and you get ADP, an inorganic phosphate that releases some energy, and that energy can be captured to take molecule A and B from outside the cell inside, or if this is the inside, take them outside, depending on whatever you need to do. You can also use a favorable concentration gradient for A to take B along for the ride. Okay. And an antiport is the same concept as a symport, but the difference is they go in opposing directions. One comes in while the other one goes out. All right. So that's the basics behind membranes and membrane proteins. I hope you understand a little bit better of what's going on here. The cell itself needs to keep its insides distinct from the outsides. So the axon, in our case, will have a bilayer that has proteins embedded within it. And, we've studied some of the voltage-gated ones, the sodium channels, in which the trigger here is the changing of the potential. I'm sorry, that was the text message there. But I hope in all that this has clarified things, and make sure you ask if there are any questions regarding anything here. All right. These are some fundamental concepts to neuroscience. We'll be dealing with membranes and channels probably throughout your neuroscience career. So make sure you got a solid foundation. Thank you, and have a nice day.